Yeah, well, it's not absolutely completely different, but as mentioned, we are in the other end. We are a preclinical company, and we are located in Umeå, and a result of the research at Umeå University Hospital. And we are not yet listed on any stock market list. But there is also another thing that differentiates us from others, and that is that Lipum belongs to one of the 2-3% of European small and medium-sized companies that have received the Horizon 2020 grant. And that is regardless of technology. So this is in competition with artificial intelligence whatsoever. And one could then wonder, why do we receive this? Well, then I would like to go back 20 years or so in time and talk about one of the founders of the company, Professor Olle Hernell. He's professor in pediatrics, so he was studying the nutrition of newborn children and the fat uptake from breast milk. And he discovered a protein, and the protein is bile salt stimulated lipase. And in fact, it is a protein that has importance for the nutrition of the newborn children. But during this research, they found that it was also present in the bloodstream, and that was not what they expected. So it turned out that everybody has this protein in the bloodstream. So they thought maybe it's secreted from the liver. So they asked colleagues to get samples of livers. And in one sample, it turned out to be very, very high level of this protein. And it turned out that that patient had fatty liver, and fatty liver is an inflammatory condition. And that was a starting point. They realized, maybe this protein has a role in the development of chronic inflammation. So they started to do studies, preclinical studies, testing, investigating the role of the target molecule, the BSSL protein, and they used genetically modified mice that don't have the protein and cannot produce the protein. And they used it first in a model called collagen-induced arthritis model, and that is a very well established, internationally recognized model for rheumatoid arthritis, that is in Swedish ledgångsreumatism. And you know, these diseases, you don't have very objective measures on this disease. You cannot measure an individual parameter and tell that you have rheumatoid arthritis. It's the physicians that have the diagnosis. That means that the disease is expressed in a kind of score. The higher score, the worse it is. And then if you expose mice to this collagen in this test, during treatment they become sicker and sicker and sicker. But if you do the same thing with the mice that don't have this protein and cannot produce the protein, they are more or less unaffected. It's so silent. I mean, you should be applauding because first indication, yes, this protein has a role in the inflammatory process. Later on, this was also studied in another model called dextrane sulfate sodium induced colitis. And this is the colon after treatment. And there you can see that the wild type mice, the colon is more or less destroyed, while the mice that are not having the protein are almost unaffected. So the approach then is that can we treat inflammation by blocking this protein? Well, looking into the mechanism, it was figured out that the BSSL protein is released from the neutrophils, binds to the monocytes, stimulate monocytes to migrate to the site of inflammation, and maintaining the inflammation. So if we can trap the protein, then maybe we can prohibit also the migration and the inflammation. But how do we trap it? Yeah, well, testing by small molecules, it turned out that small molecules cannot be that specific. So it was concluded one need an antibody that is very highly specific to bind the BSSL and to inhibit this migration. So the product that should be developed, that is a therapeutic antibody. Then using an antibody, so inducing an arthritis, and this is the pristine-induced arthritis model. Inducing the arthritis on a control, then the control animals are ill. But if they are treated, and regardless of the way of the administration of experimental antibodies, you can see that if they get the antibody, they are significantly less affected. And this is also illustrated very clearly in these photos. The control are really ill, while the animals that receive the antibody are less affected. Then looking into people having rheumatoid arthritis, and thanks to the advisory board member, Professor Solbritt Dahlqvist, we have access to patients and patient samples. And this is blood samples from patients having rheumatoid arthritis, and monitoring then the concentration of the target protein in the blood. And if we look on this side, we can see that there is some kind of correlation between the concentration in the blood and the activity of the disease. Although it's rather scattered, it's hard to see. But if you differentiate the patients, because these diseases, they have relapse and remission. And when the disease is active during relapse, you can also see that the target protein concentrations are high, while when they are feeling better, then the protein concentration is low. A similar pattern is also seen when these patients are treated with one of the dominating drugs, that is an TNF-alpha inhibitor drug, Remicade in this case. So when they are treated and the patient are feeling better, also the concentrations of the BSSL molecule is decreased. The rheumatoid arthritis market is huge. You can see that over the last 5 years, it has increased by 50%. It's around about $30 billion today. And there are several reasons for that. And because these highly specific and efficient biological drugs are used more earlier in treatments, in the treatment of them, because the price have come down. Patients are treated with a more sophisticated drug earlier in the process, which means that the market in value increases. Still these are blockbusters, but still, round about 1/3 of the patients are non-responders. They actually have no help of the drug. And another 30% loses the effect after a couple of years, which means that the physicians need to re-challenge the disease with other biologics. And at some point, there is no other alternative, and the disease will maybe have a progression. You probably heard about biologicals, the pricing of them. Here we have some of these TNF-alpha inhibitors, well-known blockbusters. And the treatment, based on the statistics from US insurances, is pricing between $20,000 to $50,000 US per year. But in some cases, like Ilaris, that is used for a rare disease, the systemic juvenile arthritis that affects children, then it's almost $400,000 in cost for 1 year treatment. And because of the background of the founders of Lipum, they have been very well aware of the medical need among children for this juvenile idiopathic arthritis, or GIA. There is a high medical need and there is a high market, despite the fact that it's a rare disease. And since it's a rare disease, it's possible to get an orphan drug designation. And that is what Lipum are aiming for as well. So you see in the treatment tree that in the first step you get some anti-inflammatory, and then you go down in the treatment tree, and when this doesn't work, then you end up in the third line where you have the TNF-alpha inhibitors, and then when they don't work, you are in the fourth line, and there you can find we have this Ilaris that is around about $400,000 per year. And Lipum are aiming for the third and fourth line treatment of the juvenile arthritis. And that means that we see the juvenile arthritis as a possibility to apply for an orphan drug designation and bring it to phase 2 studies and clinical proof of concept on our own, while other indications like rheumatoid arthritis or even psoriasis arthritis or IBD, they are far too large indications, require too much patience for be possible for Lipum to manage. So there we need absolutely partnering to be able to proceed in that direction. Okay, so here we have the founders, Professor Olle Hernell, Susanne Lindqvist, and Lennart Lundberg. And the board of directors is led by a very well experienced leader in pharma, is Ulf Björklund. We also have a very active advisory board with Rickard Holmdahl at Karolinska Institute working with the inflammatory biomarkers. And we have consultants and other partners that are necessary for our progress. And then in particular, very important, SciLife Lab. The researchers received a grant at this National Center for Molecular Biosciences that are in Uppsala and Stockholm. And the grant works in a way that you don't get the money, but you get the qualified support in the development and optimization of a therapeutic antibody. And that work has been finalized now. Still, the owners have the majority, the founders have the majority of the company. Maybe this will change now in the new share issue that we are doing right now. And at the moment, we have just selected the drug candidate. And that is, of course, a result of years of development and optimization. And now we have a humanized monoclonal antibody that will, signing contract next week, will be forward to a contract manufacturer that will have cell line development, setting up the production method. And we will get material during this process. And next year, we will have material to starting the tox studies. In parallel, we produce material for the clinical trials. That means that clinical trials can start in the beginning of 2021. And this is rather straightforward. In parallel, of course, the preclinical work will be continued, and we are especially interested in exploring this wide range of autoinflammatory, autoimmune diseases that are in this field. It's not only rheumatoid arthritis or juvenile arthritis. There are hundreds of diseases in this field that need better treatment. Okay, so how have we proceeded? Yeah, above this line we have the grants. Below we have investments. If we look backwards, initially this project the company was supported by Novo Nordisk through these Novo Seeds funds. Umeå Biotech Incubator, and of course then the SciLife Lab has been very important. But also European Union early granted a small amount, and later on then just recently 23 million SEK. In parallel, Alma Invest became shareholders. Later on the management and others have invested like business angels, and right now we have a new share issue that will close on Sunday. If anyone is interested, there are documents to fill in outside. And this looks like this. So we have a pre-money valuation of 58 million. Then we have cash, 23 from the European Union, and we will offer 30% approximately of the company. And I don't know what time it is, but I thank you for your attention. I think it's time for questions.
Thank you. Very interesting. You kind of laughed a little bit when you said that there are documents to fill in if you're interested into investing. But I know that for a fact that you have found investors through BioStock when you've been here earlier.
Yeah.
It's not a joke. It's for real.
Credit to you.
Thank you. So there are many autoimmune diseases, inflammatory diseases. Why did you specifically choose to start with this juvenile idiopathic arthritis?
Yeah, well, it is based, of course, on preclinical proof of concept. It works in these models that relates to the juvenile arthritis. That's one thing. The other thing is awareness of the medical need. And the third thing is expertise in treatment of children. So our founders, typically pharma companies don't want to do clinical trials on children and pregnant women. But in our case, we have the expertise on clinical trials on children. So it's a good combination. And you can also say that even though it's a rare disease, it's around about 300,000 patients globally. Okay, and we can only approach the third and the fourth line treatment. Still, that constitutes a market around about 2.5 billion euro. So you become a little bit surprised about that, because the pricing of the drugs are so high, like Ilara is close to 400,000. That's one thing, and there is obviously also less good alternatives.
So statistically looking at rare disease drugs, they do command usually a higher price. What are the odds of Lipum getting the orphan drug designation?
I think that is very good. I don't expect any problem with that, actually, because we have already checked that. Okay. Of course, you need it from the authorities as well. But it's very much you look into the statistics. How many have the disease? Then the other thing is that you should not apply for an orphan designation because you intend to do something else. No. The authorities don't like that, but otherwise I think it's a good chance to get it. Yes.
You mentioned earlier that you had early financing or backing from Novo Seeds, and I know the big pharma companies, they of course they are interested in orphan drug designation as well. When you go around meeting potential future partners, how important is that to them?
No, I don't think that is the most important. The most important thing is something that is extremely unusual with Lipum, and that is that we have a new target for treatment of autoimmune chronic inflammatory diseases. That is something they never heard of. In fact, when we started to actually meet them around about 2 years ago, meeting them at big pharma expertise at big pharma, they were like, never heard about it. Could it really be true?
Is that an advantage or is it an obstacle?
I think it's for a small company. I think it's an advantage initially that you can be working all alone for some time. But of course, once the results start to come, then the interest will increase. And big pharma is definitely interested. But their approach is that come back once you have the
clinical data.
No. Come back once you have the physical product. Once you have the humanized antibody that we can test in our models. And of course, that is one of the things we aim to do, to invite them to collaborate and test our antibody in other disease models than rheumatoid arthritis or IBD or so forth.
What about production? Is that a risk when you use humanized monoclonal antibodies?
No, well, I don't. I think that the development and setting up production methods today is relatively straightforward. There is a lot of companies offering these services, and the skills are today much better. So it's relatively straightforward. Okay, we can have problem with biological molecules, but you don't need that. That's not much of a risk, I think, any longer. But that's still substantial cost. So from today to have the GMP material in your hand, it's probably around about 5 million euro.
And you're right now you're doing like you said, a financing round, aiming to collect around 2.5 million euro.
Yes.
Yeah. And
we're very close.
Okay.
To have it
Close
also
to the
already.
target,
So.
yeah. How long will that take you then in the process?
Yeah, I listened to a previous presenter, and we plan. I showed it on the slide also. We plan a next round, the first quarter next year, and that is a little bit depending also because we have been in contact with around about 90 venture cap companies. We have 20 that say that they are interested in our progress, but small companies, it's a little bit too early for them. It's a little bit too small money. So of course.
Because they actually want to invest larger amounts.
They want to invest larger amounts, but they also want to wait. So for that reason, the board had a strategic decision that next year we should be prepared. So we have different alternatives. And one alternative, if we prepare ourselves for a listing at the First North or so, then we have options next year. So that is why so you could call it a pre-IPO in the beginning of next year. But I don't think we will have options. That's clear.
Okay. So then, if anyone who's here today will be interested into investing in Lipum, how would they go about it practically? Just talk to you after in the break?
And fill in the form and leave it to me. That's it. That's as easy.
As easy as that.
Yeah, of course. Then it's the board that decides who get the opportunity.
Okay.
To become a shareholder.
There's an amount of vetting involved as well. Well, Einar, good luck with the financing round and the project, of course. Yeah. Looking forward to hearing more about that. Thank you very much.
Thank you.